Hello, my dear student. Good morning. How are you? Hope you all are quite okay and fine. I am also very fine. Welcome you all to my today online class. Before going to start my today's lecture, first of all, I would like to introduce myself to all of you. My name is Mohammad Ghulam Morshed, lecturer of biology, Milestone College, Uttara Dhaka. Today I am going to discuss about some very important topic, that is the role of digestive enzymes and acids in killing foodborne bacteria. I will be discuss here the role of neutrophil and macrophage in killing bacteria. I will be discuss here the process of phagocytosis. These are the topics that will be discussed here today, and these topics are under the chapter Defense System of Human Body. Learning objectives. After completing my today's lecture, student would able to know about, or you can easily define some important definition such as neutrophils, macrophage, phagocytosis, phagocytes, diapedesis, phagosome, phagolysosome, and lysozyme. You can also able to explain the role of digestive enzymes and acid in killing foodborne bacteria. You can also able to explain the role of neutrophils and macrophages in killing of bacteria. You can also explain the process of phagocytosis in detail. Now, first of all, I would like to discuss about the role of acid and enzymes of digestive canal to kill bacteria of food. Look at the figure here. This is the simple anatomical structure of the stomach. You see here some gastric glands. Gastric glands are usually present at the inner lining of the stomach. Now, first of all, I would like to discuss about the role of digestive enzymes and acid in killing foodborne bacteria. As you know that bacteria are the most common causes of food poisoning. Normally, we intake different types of food in our daily life. During the ingestion of the food, some foodborne bacteria such as E. coli, Vibrio cholerae enter into our digestive tract, but our body has several ways to check foodborne microbes. Stomach acid and digestive enzymes not only helps in digestion but also kill some bacteria, particularly if the food is lightly contaminated. Sometimes we intake some contaminated food, so there is a possibility to have some waterborne disease or foodborne disease. And however, the bile and mucus of alimentary canal also plays some very important roles in killing of bacteria. Now, first of all, I would like to discuss about the role of digestive enzymes in killing of bacteria. There are different types of digestive enzymes secreted from the digestive tract. These are the lysozyme enzymes, pancreatic protease enzymes, and lactoperoxidase enzyme. The lysozyme enzyme is a very important enzyme secreted from the salivary glands, kills bacteria that bring with food. Lysozyme is an enzyme that are capable of breaking down the cell wall of some gram-positive and gram-negative bacteria. So, lysozyme has strong bactericidal effect. Bactericidal means the agent that can kill the microbes directly. So, lysozyme has a strong bactericidal effect. Now, another very important enzyme that is secreted from the pancreas, this is called the pancreatic protease enzyme. Pancreatic protease enzyme is important in killing of harmful bacteria in small intestine. Proteases tend to have a synergistic effect with antibiotic, making the bacterial control more effective. In case of various types of infection, we intake antibiotic to treat the infectious disease. But protease enzyme, which is secreted from the pancreas, can have a synergistic effect with the antibiotics. What do we mean by synergistic effect? Antibiotic is actually, in case of synergistic effect, the antibiotic increases the effectiveness of the protease enzyme. Similarly, the protease enzymes is also increase the effectiveness of the antibiotics. So you can easily understand that if 2 plus 2 equal to 4, but in case of synergistic effect, 2 plus 2 equal to 5. Another very important enzyme that is secreted from the salivary gland and mucus gland, which is called the lactoperoxidase enzyme, that acts as a natural antibacterial agents and kill gram-positive and gram-negative bacteria. Now let's discuss about the role of stomach acid in controlling or in killing foodborne bacteria. As you know that the hydrochloric acid is produced from the parietal cells of the stomach, that actually responsible for making the acidic environment in the stomach and also responsible for killing of bacteria. Number 3, the mucus. Mucus normally lines most of the white surface of the body, including the respiratory and digestive tract. Typically, mucus contain a number of types of antimicrobial compounds, including lysozyme enzymes and secretory antibody, immunoglobulin A, which function primarily by binding microorganisms and thereby preventing their contact with the host tissue. Number 4, the bile. Bile secreted from the liver, then is stored in gallbladder, poured into the small intestine. The main functions of the bile is digestion, but the bile salts are mainly two types, sodium taurocholate and sodium glycocolate, are capable of killing bacteria. Now let's discuss about the role of neutrophil in killing of bacteria. First of all, you have to know about the neutrophil. As you know that the neutrophil is a granulocytic white blood cell. Let's see the figure or structure of the neutrophil. Neutrophil is a granulocytic white blood cell, and it contains a lobed nucleus. The nucleus may be 3 to 7 lobed, and the cytoplasm contains lots of numerous secretory granules. Neutrophil in killing bacteria. By the three way neutrophil can kill bacteria: by the phagocytosis process or degranulation and net formation. Phagocytosis is the process by which neutrophil can engulf and destroy any foreign pathogens, but degranulation is the process by which the secretory granule becomes broken down and release some important chemical compounds that is responsible for killing or damage the microorganisms, and net formation is another way of trapping or killing of microorganisms. Now the role of neutrophils in killing of bacteria. Neutrophils are normally found in blood stream and the most abundant type of phagocytes, consisting 50 to 60% of the total circulating white blood cells. Neutrophils have lysosome that can contain the various bactericidal and the digestive enzymes which can destroy bacterial cells after engulfment. Once they have received the appropriate signal, it takes about 30 minutes to leave the blood and reach the site of an infection. Once the neutrophil enters the tissue, it never returns to the circulation. They are ferocious eater and rapidly engulf invader coated with antibodies and complement or damage cells or cellular debris. Neutrophils are capable enough to engulf and destroy any free moving microorganism, and sometimes they are capable enough to engulf opsonized bacteria or microorganism. Opsonization is a process by which microorganisms are more susceptible to phagocytosis. Some bacteria are coated with antibody and moves toward the phagocytic cells, so that it is very easier for phagocytic cells to engulf and destroy the antibody coated microorganism, and this process is actually known as opsonization. And sometimes complement proteins may be activated, and they can also play a very important role in phagocytosis process. Now the neutrophil do not return to the blood and turn into pus cell and die. Neutrophils can secrete products that stimulate monocyte and macrophage to phagocytosis of IgG antibody-coated bacteria. Their secretion increases phagocytosis and the formation of reactive oxygen compound involved in intercellular killing. Now let's discuss about another very important topic, that is role of macrophage in killing bacteria. Look at the figure here. This is the phagocytosis process. How a macrophage engulf and destroy any foreign pathogens or microorganism. This is also a phagocytosis process, and the macrophage engulf the bacterium and destroy it. The role of macrophage in killing of bacteria. Macrophages are large white blood cells. These arise from bone marrow stem cells which develop from the monocyte that are released into the bloodstream. First of all, you have to know about what is macrophage. Macrophage is actually the differentiated mature monocyte. As you know that monocyte is a agranulocytic white blood cell which is originated from the bone marrow, and after maturation it moves through the blood to the site of infection. When it reaches in the site of infection, it becomes larger and form and give rise to the macrophage. So macrophage is the larger size which is actually derived from the monocyte. The monocytes are actively phagocytic and bactericidal. Within 2 days or so, the bloodstream monocyte immigrate into tissues where they settle down, enlarge and become fixed macrophages which also have phagocytic potential. The total pool macrophage is referred to as the system of the mononuclear phagocyte. The system is scattered throughout the connective tissue, basement membrane of small blood vessel, liver, spleen, lungs, lymph nodes. Now, actually macrophages are of mainly two types: mobile macrophage and tissue macrophage. Mobile macrophages are those which are derived from the monocytic cells, and a monocytic cell is actually give rise to the mobile macrophage. And there are some tissue macrophages are also found which are reside in the different types of tissues like spleen, lungs, lymph node as a permanent resident. Now the role of macrophage in killing of bacteria. Macrophages are more active in phagocytosis than monocyte and develop many more granules containing hydrolytic enzymes. They are actively phagocytic and will engulf and destroy foreign particles such as bacteria. Additionally, macrophages produce important immune proteins called cytokine, which are hormones that deliver messages to other cells of the immune system and alert them to the infection site. Another very important functions of the macrophage is it also act as a antigen presenting cell. When macrophages engulf any foreign pathogens or microbes, it degrades the microbes into very smaller size, which is also act as an antigen, and this macrophage can display these small particles or antigen over the surface of the macrophage so that it can very easier to detect or recognize by especially T lymphocytes or T cells. So the macrophage is also act as an antigen presenting cell, and it also involved in the cell mediated immunity, that is T dependent immunity or cell mediated immunity. Now let's discuss about the process of phagocytosis. Look at the figure here. This is the very simple process of phagocytosis, and before going to discuss the phagocytosis process or to understand the phagocytosis process clearly, you should have some idea regarding what is phagocyte. The phagocytes are some cells which are capable of ingestion and destruction of microorganisms. The cells are the phagocytes. Phagocytes are of mainly two types: neutrophils and macrophages. And what is phagocytosis? The engulfment and destruction process of microbes by phagocytes called phagocytosis. Now let's discuss about the process of the phagocytosis. Neutrophils and macrophages perform these functions through several continuous steps. Actually, phagocytosis process have several important steps. Now let's discuss about these steps briefly. First stage is called number 1 stage, the delivery of phagocytic cell to the site of infection. The delivery of phagocytic cells, monocyte or neutrophil, to the site of microbial infection involves two processes. Number 1, diapedesis, and number 2, chemotaxis. Diapedesis is the migration of cells across cellular vascular wall, which is initiated by mediator of inflammation, histamine, prostaglandin, etc. Diapedesis is the process by which phagocytic cells cross the capillary membrane and reach at the site of infection. But how can it possible? When any injury occur or when any inflammation occur in our body, the inflammatory cells can secrete some chemical substance like histamine and prostaglandin. This chemical substance, especially histamine, are responsible for increasing the permeability of the capillary wall so that it can very easy for the phagocytic cell to move toward the infection site through the capillary membrane. And this process is actually called the diapedesis. And the chemotaxis is another very important process by which a phagocytic cell can move toward the site of infection. Chemotaxis means the movement of any organisms or any cells in response to the chemical stimulation. As you have already know that when any injury occur or any inflammation occur in your body, the inflamed cells secrete some chemical substance. Not only inflamed cell but also infectious agent can also produce some chemical substance which act as a chemotactic stimulant, and macrophage and phagocytic cell is actually attracted by this chemotactile stimulant and moves toward the site of infections. And this process is actually called the chemotaxis. So the chemotaxis is even cell concentration of phagocytes at a site of injury results from the chemotactile response by phagocytes. Our second step of phagocytosis is the phagocytic adherence. Phagocytosis is initiated by the adherence of the particles to the surface of the plasma membrane of the phagocyte. Phagocytic adherence means the attachment of the microorganism to the phagocytic cells, and this is called the phagocytic adherence. And number 3, the ingestion. After attachment of the phagocytes to its target, some sort of signal generation, which is poorly understood, result in physical or chemical changes in the cells that triggered ingestion. Actually, ingestion is an engulfment process that involves unfolding or invagination of the cell membrane, enclosing the particle and ultimately releasing it into the cytoplasm of the cell within a membrane vesicle. The end result of ingestion is entry of particles enclosed in a vesicle derived from plasma membrane of the cell. This structure is called phagosome. This is the very important terminology. You have to know about this. What is phagosome and how can a phagosome is formed during the process of the phagocytosis? Ingestion process is very simple. You can easily understand that. When any microorganism attaches to phagocytic cell, the cell membrane of the phagocytic cells are extended around the microorganism, and when the two sides of the extended portion of the plasma membrane fuse together, the trapped microorganisms are now penetrated inside the cell and enclosed in a vesicle which is actually derived from the plasma membrane of the cell, and this is actually called the phagosome. And the fourth step is very important, that is the formation of the phagolysosome. The phagosome migrate into cytoplasms and collide with lysosomal granules, which explosively discharge their content into the membrane enclosed vesicle, which is called the phagolysosome. Membranes of the phagosome and lysosome actually fuse, resulting in a digestive vacuole called the phagolysosome. The formation of the phagolysosome is very important. how a phagolysosome is formed during the process of the phagocytosis. This is a very simple process. When phagosome migrate into the cell, as you know that in the phagocytic cell there are lysosome are present, and when the lysosome get attached to the phagosome, their lysosome contain different types of hydrolytic or proteolytic enzymes, and this proteolytic enzyme is dumped into the phagosome, and ultimately a phagolysosome is formed by the fusion of phagosome and lysosome. And now the number 5 step of phagocytosis process, this is called the intracellular killing of organisms. By 10 to 30 minutes after ingestion, many pathogenic and non-pathogenic bacteria are killed, followed by lysis and digestion of microbacteria by lysosomal enzymes. Actually, lysosome contain different types of proteolytic enzyme, and this proteolytic enzyme is actually responsible for killing of microorganism. Not only this lysosomal enzyme, but also some secretory granule contain some toxic chemical substance that also responsible for killing of microorganism. And the 6th step, the intracellular digestion. Dead microbes are rapidly degraded in phagolysosome by various hydrolytic enzymes, including lysozyme, protease, lipase, nuclease. Neutrophil die and lyses after extended phagocytosis, killing and digestion of bacterial cell. Macrophage ingest digested debris and allow insertion of microbial antigenic compounds into the plasma membrane for presentation to lymphocyte in the immunological response. Actually, we have already discussed about a little bit about how macrophage act as a antigen presenting cell. When any pathogen that is already killed by the various types of lysosomal enzymes, and the killed microorganism then degraded by some hydrolytic enzymes, and these small particles of this microorganism act as an antigen, and macrophage can able to display this small particle or antigen over the surface of the macrophage cell membrane. But in case of neutrophil cannot able to display their antigen over the surface of the neutrophil cell surface. So neutrophil does not act as an antigen presenting cell. Now the homework, write down the process of phagocytosis. Now creative questions. During ingestion of food, many foodborne microorganisms can enter our digestive tract, but most of the microorganisms are being neutralized in gastrointestinal tract by various chemical barrier. Besides, these two special type of blood cell can engulf and destroy the microorganism by special destruction process. Number A, what is lysozyme? B, what do we mean by phagocytosis? C, describe the process of neutralization of phagocytic microorganism in gastrointestinal tract as mentioned in above stream. D, analyze the process of engulfment and destruction of microorganism by two specialized cell as mentioned in above stream. Thank you for your patience hearing. I think today's lecture is very important lecture for all of us. I am requesting you all to watch my video lecture very attentively so that you can understand each and every points that will be discussed here today. If you have any queries regarding my today's lecture, don't hesitate to communicate with me. You can ask me regarding your queries, or you can ask your respective subject teacher regarding your queries. We all are prepared to solve your problem. Thanks you once again.